Yeah, I never really got it. You give them so complicated. I play.
Well, I can't do it. Okay, sorry, now we start. So cells make up everything. There we go. So the first thing you need to know about something called cell theory. And there's 3 parts of cell theory. The first one is that all living things are made up of cells, whether it's a single cell or whether it's many, 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 many cells. All living things are composed of cells. The cells are the basic unit of life. It's the lowest in terms of the organization of things in biology. We talked about a long time ago. The cell is the one that has that you can be considered living. An organelle by itself is not considered living, but a cell is. The third part of cell theory is that all cells come from pre-existing cells. Now, you can go back and be like, well, where did the first cell come from? We're not talking about that right now. But from that first time, then everything from them came from that one cell. So that's that. What kind of cells are these? Plant cells, good. And this is an animal cell with contractile vacuoles. Look at those, they're cool. So that's cell theory. I'm pretty sure that was in your book. You learned this in Bio 1 as one of the standards. I'm going to go with that. Okay. So one of the things I like to talk about is that, like, you are made up of lots and lots of cells. Do you know that every single cell in your body has the same DNA? Did you know that? Yeah. So why do you, like, does your skin cell do the same thing as your liver cell? No. But they have the same DNA. Right. So only part of your DNA is used in every cell of your body, and the rest of it is turned off. And sometimes it's turned off completely, all the time, forever and ever and ever and ever and ever. And that's what it's called cell differentiation. And it happens early on during development. When you were first conceived, you were called a zygote. So when sperm fertilizes egg, that forms a little zygote. And that is, I don't have a zygote. I don't think you make zygote, but imagine. That cell is totipotent. It can become anything. It's a true stem cell. And early on during development, all those cells are stem cells. They can become anything, because their DNA, the cells haven't differentiated yet. But at some point during development, differentiation starts. And at that point, DNA started to turn off. And then from then on, it can only be a certain type of cell forever and ever and ever. Your liver cell will only ever be a liver cell in your body. Now, scientists now can take cells out of you and actually can take them back and turn them back into stem cells, which is really cool. And it's pretty much kind of taken away the need for like embryonic or fetal stem cells. There's still a lot of controversy in that area, and some people still think we need fetal stem cells, but they can take your own cells and turn them backwards. But they have to expose them to a lot of different chemicals and things like that. Anyways, we'll get to that later. But so stem cells are things that can become anything, any type of cell. The only stem cells you have right now in your body, are called hematopoietic stem cells. Have we talked about that word yet? Okay, not in this class. Okay. Hematopoietic stem cells are the ones you have right now in your bone marrow. And they can become any type of your blood cells. So they can become red blood cells, any type of white blood cells, and they can become platelets. Platelets aren't really cells, they're fragmented cell pieces that are used for blood clotting. But that's kind of what they're showing here. So they're not true stem cells, and that these stem cells can't become like a skin cell. They can only become any type of blood cell. So it's happening all the time. Those cells are constantly dividing. Your red blood cells only survive for about 120 days, and then your liver breaks them down, and you're always making new stems. You're making new red blood cells. And a lot of times, some of your white blood cells, there's all different kinds. Some only live for like 3 days. Yeah.
Would it be easier to make like regular stem cells than taking just another like taking a stem cell and making it?
Not necessarily.
Okay.
Yeah, not necessarily. So, yeah.
Why is like stem cell research such a problem?
What do you mean a
I
problem?
like a lot of them are against it.
Well, that's because they reduce fetal stem cells, embryonic stem cells. And they would, I need to block that cabinet, it's really annoying. Sorry. Sorry, so, yeah, anyway, because they would take, a lot of times okay, we go over here. We'll talk about this later, but I might as well talk about it a little bit now. They would take an embryo that's been fertilized. Like a lot of times, women that have to undergo fertility, they might have to do something called in vitro fertilization. While their eggs are harvested, they have to get a sperm sample, and then they fertilize it in a test tube, which is called in vitro, right? And then they do that a lot of times, and then they would freeze them, okay, or store them for when the mom's like ready or whatever. So right now, there's a lot of frozen or stored embryos that when women are finished like having their kids, like, and they don't want anymore, they're just locked away, like they're put away. They're not, like, according to law, they're not allowed to be destroyed. So they're just sitting in these banks of embryos. And scientists would want to use those because they're never going to be used. They're just going to sit there. So you might as well use them for research purposes, but there's a lot of controversy, like, well, that could be a person, and should we do that? And well, you know what I'm saying? Like, there's all this. So that's why. Now they think they can take stem cells from like the umbilical cord after a baby's born. They can use that for research. But the controversy lies in like, is that a human and is that ethical and whatever? And some people would argue yes, and you shouldn't do that. And some people would, and scientists, some scientists would argue. And some scientists would say what I said just now, which is that, if they're never going to be used, we might as well use it for some purpose, to advance science. So there's two sides. And we will talk about that later on in a different chapter, but that's why. But so scientists can now take your own cells and take them back and turn them into stem cells. So is there really a need for those anymore? Some scientists say no. So that totally eliminates that controversy. You know what I mean?
Are those stem cells just do they take that? Or are they just as effective as the cancer?
I think so. The research is still relatively early, but they do so. So but I heard a thing on NPR the other day that there were actually scientists talking about this. And like this one lady said, yes, it is just as effective and we shouldn't use this. And this other scientist said, no, there's still a need for fetal stem cells to do certain things. So yeah, there's still a lot of arguments. Yes.
Something is aborted, gives it slow sometimes for themselves.
Yes.
Yeah.
I think
I think we reminded me. I felt a little weird about that.
For your knee? Oh, well.
I felt a little weird about that.
Just know it wasn't your decision to do that.
Yeah.
I mean, you know what I mean?
Mhm.
So it is kind of strange. I have, you might have your own personal beliefs about it, and I know I felt a certain way, but then after I had kids, I feel like, this is just my opinion, but after being pregnant, like when I was pregnant with the twins, I had 2 in there, and the doctor like talked to me about there's certain genetic testing they can do early on, and to determine if your children have, some sort of disorders. And when they do these ultrasound, scans, and when they do that, sometimes they can detect abnormalities, and then they can offer you these tests, like, do you want to do these tests or whatever. And Fletcher and I talked, and it was funny, before we got pregnant, we were like, we would want to know everything there is to know about them to prepare ourselves, to just know what we have in store for us. But after we did that, and they were like, they do this blood testing if we wanted it, but they told us that there was no way they'd be able to know which twin it was. And, and we were like, do we really want to know? Like, no matter what, like, our minds completely changed. We were like, it doesn't matter to us, like, when I was pregnant, like, no matter what it is, we wouldn't want to do anything about it, so why even worry about the testing? It was really weird, because I really felt strongly one way, and then after I got pregnant, I really changed my mind. It's really weird. I never thought I'd be like that. So, personally, after being pregnant, I don't think I could ever do anything like that, no matter what, you know what I mean? But I also don't know, like, I don't know, it's just very, very strange. But it wasn't your decision.
Oh, no.
So, I mean, it is strange. I think I would feel weird about that too, but
Yeah.
you didn't decide to do that.
Yeah, they said like they could do that, or there's another thing like they would have like just taken the cells out,
Yeah.
or taken like cartilage from another part and then grown it
And do that, yeah.
and put it back in without it taking
That's
a couple
interesting.
months.
Huh.
So they're like
It would take a long
it
time.
would take like I'd have to
Yeah.
go for one surgery, wait like another
Yeah.
month or 2 months. months.
Yeah, yeah.
Go back and redo it.
Yeah. Huh. Just do it now. Yeah, yeah.
Just do it over now.
It's crazy. Sorry, that's a whole nother conversation. But, anyway. But, so anyway, sorry. We will talk about this when we do chapter 10, 11? Anyway. Okay, so moving on. And this is also, see, biology's so fun. I also read something the other day about how this guy was like, viruses are alive. Like I read the scientific article that said viruses are alive. And they think viruses, somebody mapped in the evolution of viruses, like the tree of virus evolution. It's crazy. Like, I don't know. I've always thought the viruses aren't alive, and now I'm gonna have to really, anyway, things are always changing. But anyways, what does it mean to be alive? Right now, what most scientists think is that what it means for an organism to be considered alive is that they do all these things that are necessary for life by themselves. So one, they have the ability to reproduce on their own. They have the ability to grow and develop, respond to stimuli. All organisms, no matter what, are open systems. We've talked about that. They exchange things with their environment. They have some sort of metabolism. They excrete waste products. They can die. They have some sort of genetic material, which allows them to be able to pass that on to their offspring. They maintain homeostasis and they have the ability to regulate. What's interesting about viruses is that they can't do all those things by themselves. They have to rely on a host to do a lot of those things. But we have a whole chapter as well on viruses. So we will talk about that when we do that chapter. Isn't it exciting? Sure. I'll be excited for it. No problem there. Okay. So, how do we study cells? So the next part of this chapter is all about microscopy. So microscopes are how we study cells. Robert Hooke was the first person we call a cytologist. A cytologist is a biologist that studies cells. 1665, it's a long, long time ago. But it was cool. Like he developed this microscope and he looked at all the, and it's basically a glorified like magnifying glass really at that point. But he looked at things like this is a flea. And like he drew all these pictures of what he saw and he wrote this book. Physiological description of minute cells. And like that's what this book, it was like a bestseller. People are like, that's what a flea looks like? Like all they can imagine these little things hovering around like on their jaws or something. They're like, whoa, I mean this was like a huge thing. Now we're like, whatever, you can look up, you know, it's not a huge deal. But anyway, so we use microscopes now for all kinds of stuff. You will use light microscopes in this class. Some of you, someday, if you haven't already, will use electron microscopes. Well, let's talk about them. So there's a diagram in your book, looks like this. And here we can look at the top. This is stuff you can see with your eyeball by itself. So you can see the height of yourself. You can see neurons, actually. I mean, you can't see yours, but you could see one if you dissected a human. A neuron is something you can actually see. They're very large. A chicken egg is analogous to like your egg, but it's a really big egg. Ostrich eggs are, I think, the largest one. Frog eggs are pretty small, but you can still see them. Then you have to move into the area where you see smaller things, but you can use a light microscope. Things that would be a millimeter to maybe a micrometer. The little U thing is micro, remember. These are things like most plant and animal cells you can see. You can see really large organelles like the nucleus. You can see most bacteria, bless you. Not all bacteria. And maybe with some really, really, really good light microscopes, you can see a mitochondria, but most of like the ones we have, you can't. But light microscopes, why they're advantageous is because you can look at living organisms. You don't have to kill them to look at them. The electron microscopes, a disadvantage of those is that specimens have to be dead. But the way the light microscope works, so I have. Yeah, you have to coat them with gold. Hold on. Hey, sweetie. Oh. Okay. Great. Look at you. Okay. Yes. Okay. I can go to the gym already. Okay. Oh, you should go. You should go. Good. Yeah. It's fine. Well, yeah. 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 I love you. That's great. Love you. I'll call you later. You okay? Okay, well, I love you, sweetie. I'll call you later. Bye. Sorry. See, their game has been moved to tonight.
Yeah, our
Oh, okay.
we're moving our homecoming festivities to next week.
Yeah, and he's moved all his to the 16th. Sorry, Fletcher. He gets emotional. He's like a female sometimes.
I told my dad that. I was like, you're
Whoa.
It's really funny.
I don't know. I like to do the yard work. I mean, we're like reverse roles in our heads. Weird. He talks about his emotions a lot. I don't. It's very strange. Sorry, he just got off. Anyway. Okay, moving on. The light microscopes. 0.2 micrometer resolution is about the best you can do. Not ours. You can look at live things. So you have visible light that passes through the specimen, and then it's magnified, and you can see it, and it's exciting. You can even take videos of your organisms moving around if you wanted to, or pictures of them. But electron microscopes are way better at seeing things that are much, much smaller. They originally developed in the 50s, which is a long time ago, but you can see down to like 2 nanometers, sometimes even smaller than that these days. Over 100 times better than the light microscope. You can look at all the organelles, even with the best electron microscope, you can even see atoms, which is really cool. The only disadvantage is that your specimen has to be dead, and it has to do with how you prepare it, and I'll talk about that with the different types. Has anyone ever worked on one? Yeah, I did a lot of work one summer in college at the School of Medicine. I was working with 2 things. I was studying fetal alcohol syndrome. I was injecting, this is kind of sad, but I was injecting fertilized chicken eggs with different concentrations of ethanol, and their incubation time was like 21 days. And then at different times during the incubation period, I would basically kill it. Some were really early on, and I would just you could see the big yolk sac, and you could see all these blood vessels and a little tiny embryo, and I would have to take it off. And I was actually looking at heart development, so I'd have to dissect it, and anyway, it's cool. But sometimes I was also looking at development of the retina and seeing how fetal alcohol syndrome affects that. I have to kill them at like day 20. It was bad. It was like an entire chicken in that thing, and I have to rip their heads off. It's awful. Yep. And I have to dissect their eyes, but I used transmission electron microscopy, because then we would put their eyes in this agar block and slice it real thin and look at it. It was really cool. But
Pardon this interruption. Due to the excessive inclement weather predictions, the varsity football game versus White Knoll will be played tonight here at Spring Valley instead of tomorrow. Our varsity football players are reporting to the varsity locker room at the beginning of lunch. The B team and JV games at White Knoll tonight have been canceled.
Okay, all homecoming activities are gonna be next week, starting Thursday. So bonfire Thursday, and then pep rally Friday, and wear your homecoming shirts to the flag weekend.
So there's no blackout tomorrow, so there's.
Well, you can still go back out, but it's gonna resume next week as well.
Can we do another drink?
I know. I want another dressing. My friend Jean's every day. Okay. Anyway, usually electron microscopes are in the basements of buildings because they're very sensitive to vibration. So basements would be underground. They'd be sort of shielded from those vibrations. They're also very, very expensive, like millions of dollars. So only certain people that have been trained should be allowed to use them. Anyway, they're really big machines, and they're usually connected to computers. This is obviously a really old picture because that's an old school kind of thing. But you can cause different parts of them to fluoresce, and you can look at different pictures. I'll show you some stuff later on, but this is kind of what it looks like. I mean, this is really big. Your sample would be like way down here. This is where the electron beam would be located, like going down there. So there's two types, transmission and scanning electron microscopes. So a transmission electron microscope, or TEM, is used to study the internal structures of cells. And what happens is that, like I said, the way you prepare the specimens, you put it in some sort of agar and you put it in this machine that makes these really small, like really thin slices of your specimen. And then you take those and obviously you don't kill it, okay? And then you take the slices and put it on like a thing and put it in the microscope, and you can see the inside structure. So the organelles, this picture showing a rabbit trachea, and these would be villi that kind of go, you know, you think of like this. They beat upwards. Have we talked about this? They beat up, and you have it too. They beat up in your trachea. And what you like, you have snot and stuff, and basically it beats up to get all the crap out. And like you have a loogie, like you're getting all that phlegm out, and you can either swallow it or spit it out. But it's helping to trap all the particulate matter before it gets in your lungs. And it beats away if you take it away from your lungs. Anyways, so we have that. This over here is showing bacteriophages, which are these guys, viruses, and their surrounding in the middle is a bacteria in the middle. So see all those viruses surrounding that bacteria? Bacteriophages are viruses that only infect bacteria. They don't affect you. I mean, they don't infect you. Anyways, they're really cool viruses. Antibiotics only work on bacteria.
So are they made up of bacteriophages?
No. That's a good question, but no. They're made up of different medicines that will attack their cell walls and make them more susceptible to your immune system, or they can affect their metabolism or something and kill them. We have a whole chapter on bacteria as well we'll talk about. We have a whole chapter on like everything. We have a lot to do in this class. So the electron beam is aimed through the very thin section of your specimen here. And there's electromagnets in there that help to focus the image. And the way the electron beam is like bounced off the different parts, you will show different things in your pictures, different densities, so darker versus lighter, stuff like that. So sometimes you can even stain certain sections or parts of your image with heavy metals to cause the picture to be enhanced in certain ways to see particular structures. Now look at, okay, stare at this picture here, stare at it, and look at it there. That's a rabbit trachea, but you're seeing something very different, okay? So this is the image you get from using a scanning electron microscope, or SEM. So scanning electron microscopy allows you to see the outside or topography of whatever you're looking at, okay? We say TEM is used to look at ultrastructure, or the inside structure of a cell. SEM is used to look at the surface, okay? So these are the microvilli on the rabbit trachea, so you can see the outside, okay? This is a porcupine quill. No wonder it hurts when you pull it out. Look at all those little barbs that will like rip your skin when you try to pull it out. Here's a flea, not the same type of picture that Robert Hooke drew. But the way you have to prepare your specimens here is you coat them with a very thin coating of gold, and then the electron beam will excite the gold atoms on there. Depending on how tall they are and where they are on there, it'll produce different vibrations, and the computer will develop that into an image that you see. You see great depth of field. It looks almost like a three-dimensional picture. You can, I mean, if you ever care, you can Google just SEM pictures and get all kinds of really cool pictures. Here's an ant. These are fish gills.
Like one bump and then
I don't think it's that way, but.
No, I don't.
There's a bacterium, actually two of them. Ooh, there's a red blood cell. Look at that. Oh, this is cool. So this is showing a bacterium being engulfed by a white blood cell. Canidia 4, you don't know what that is. This is an organism undergoing mitosis, basically it's budding off. I know. This is the eyeball of a fruit fly. The Staphylococcus melanogaster eyeball.
Look what I found.
Have you guys seen, there's a Twitter page that has really cool pictures of like, it's like stuff like that, and it has like pictures of like your taste buds.
This is a mouse red blood
They're
cell.
really ugly though. Like your taste buds are serious.
Yeah. Frozen and fractured red blood cells in a mouse venule, like a vein. That's crazy looking.
Can we look up our taste buds?
Taste buds.
Are those things or they look like that?
They can, they're stained. They don't really look like that.
Yeah, they look beautiful. They look really awesome. Yeah. I know, wait, yeah, that one, the one, if you go down, go the one under it.
This one?
To the right too.
This
That
one.
one, yeah, that one's better.
The human tongue. You have different types of taste buds. They all kind of look different depending on where they are on your tongue. They do different things. You know, they can taste salty, sweet, sour, umami. So they look differently depending on the type that they are. Anyways, okay, moving on. So how do we study cells? Another way you can do it is to isolate organelles and study cells. So a way to isolate organelles is a process called cell fractionation. And basically what you do is you take your tissue sample, which you have down here, and you homogenate it. So you put it in a blender, basically, and go, and it like breaks up the cell membrane, and all the organelles kind of spill out into this like homogenated stuff. And then what you do is you use a centrifuge, which is a thing that spins really, really fast. You heard of a centrifuge before?
Yeah.
Okay. And it separates things based on density. So if you spin something really fast, the thing is the heaviest or the densest will go to the bottom. Okay. So you can spin it, notice here, at different speeds for different time frames. So 800 Gs, 20,000 Gs, 100,000 Gs, 150,000 Gs. That's really, really fast.
And they do like this to like separate blood.
Yeah,
Like,
that's
in
exactly
the
what they
cells,
do.
But I mean, like today, so this is like.
Same thing.
It's like, so this is like even faster, so they're gonna separate.
Yeah, ultracentrifuges are really, really fast. A million times like the force of gravity. They can go up to that.
How does that even spin that?
Yeah. Whatever. All right, look. So 800 Gs or 10 minutes, you get really big things. So the largest organelle in the cell is the nuclei. Obviously, and you get some cellular debris, and then that would be at the bottom. It forms something called a pellet. Has anyone ever done something like this before? It really does form like a pellet. It looks kind of whitish at the bottom, and then all the liquid at the top is called the supernatant. Okay, and then what you do if you're like, okay, I don't care about that nuclei, but I want something else in here. You pour the supernatant off into another test tube, and you can just discard the little pellet at the bottom. Okay, it's not like hard; it's kind of liquidy, but you can actually see it at the bottom. And then if you wanted to find like a ribosome, let's say. You want to spin it even faster for a longer period of time. Here you'd have the next most dense would be things like mitochondria or chloroplasts, stuff like that. We're looking for a ribosome that wouldn't be in our pellet yet. So we just pour out the supernatant again and spin it again and discard whatever is in there. At this point, you probably would get pieces of the cell membrane. Ribosomes are tiny, tiny, tiny. You wouldn't get them yet. And some other larger things, vesicles maybe. So your ribosomes would be in here. Spend a last time there, you get the ribosomes, and you can do whatever you want to do with it. Discard the supernatant. But that's called cell fractionation, how you get the different organelles and isolate them, and you could do whatever you wanted to with it. And that's that. So they can do things like look at what enzymes are present in the pellet or different proteins, stuff like that. They could even maybe look at the pellet if they want to look at what mitochondria looks like under an electron microscope. Take that pellet somehow, get it ready, and look at under the electron microscope, depending. This is what a centrifuge would look like. Sometimes they're big and they can fit like they're maybe the size bigger than like a microwave. They would fit like on a lab countertop. Some are really big, but most of them aren't that big. So like I said, they can be even over 1 million Gs. Microcentrifuges are used to do what we call biotech stuff, which is working with DNA, RNA, proteins. Really, really small things. Microcentrifuges are relatively small. You can see one here. They would be like half the size of the desk, maybe this tall. They're pretty little, and you would use things called microtubes. I'll show you. Like in big centrifuges, you use regular test tubes, regular sized glass test tubes. Microcentrifuges, you would. You would use tiny test tubes that look like this. They even have ones that are a little bit smaller than this. I have a really, really small centrifuge. It doesn't go very fast. But I'll show you how it works. Whenever you're working with centrifuges, you have to make sure that you balance it out. Has anyone ever done one? Like a real, you know what I'm talking about? You balance it out. Imagine you're working with glass and you just put one in here, okay? And you're allowed to spin. It's just like in a washing machine. Have you heard your washing machine go bong, 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 and then stop? Okay, it's because like when you open up, you're like, oh, all your clothes are like at one side. And when it spins, it's uneven and makes it go all wonky. Same kind of thing in a centrifuge. If your sample's just in one test tube and you want to spin that one down, you can't just put one in there, especially if you're working with glass in a big centrifuge. You have to put like water in another one and put it on the opposite side. So when you spin it, it would keep it all even so it doesn't get all crazy. It doesn't matter so much in something like this where there's a really small and whatever. The ones that matters in are the big centrifuges where you're working with glass because it could explode in there and you have a mess. And if you're trying to do blood like this. So, anyways, you just turn it on and go. Ooh. Hey, this is like a really small, it doesn't go that fast, but you spin it for a certain amount of time and then let it stop and take it out and do whatever you're going to do with it. So, no. We use these. We don't use it. Use what?
The centrifuge at the vet clinic, it like, the thing that holds the test tubes in, it like broke while it was spinning. And so there's just like blood everywhere. And it looks like someone like tried to murder this.
Oh, yeah. And you use it a lot in diagnostic stuff. Doctors' offices have centrifuges, veterinary clinics, yeah. That's gross. The blood in a centrifuge is gross.
Is that what you do with like blood sample?
Yep. Yep. What did they do with it? Exactly what they do. Okay. All right. So let's go on a fast tour of the cell. Ooh, okay. So you need to know all cells, and I mean like all cells, prokaryotes and eukaryotes, all cells, okay, have a cell membrane called the plasma membrane. It's a bilayer, it's the focus of chapter 7. But all cells have a plasma, otherwise known as cell membrane. All cells have stuff called cytosol. Cytosol is that jelly-like substance inside the cell. Cytosol. The cytoplasm, look, is the cytosol plus organelle. So you might not have learned cytosol before, you might have learned cytoplasm, but actually cytoplasm is the organelle plus the jelly-like stuff. But the jelly-like stuff by itself is called the cytosol. All cells have some sort of genetic material, generally DNA. and they form chromosomes, or a chromosome. Bacteria usually only have one chromosome. Eukaryotes have multiple chromosomes. And all cells have ribosomes. Ribosomes are the tiny, tiny, tiny things. You can call them organelles, sort of. Put them in quotations because they're not the conventional organelle that we think of. But they're the site of protein synthesis. The messenger RNA will go to the ribosome to be translated to make a polypeptide. You don't know what I just said. Okay. We have a couple of chapters on. Right now, you should know it's the site of protein synthesis. Fancy on me because there's no space for bells. They're a little too big for me. I got them in Boston, what, 2 years ago when we went. You can go with us. I'm sorry. Are you gonna go? Yeah, yes, I'm going this year. So I went when Marie was like an infant and Fletcher wanted to kill me. He was like, "You're not going next year." And so I didn't go last year, but this year I was like, "Fletcher, I'm going. I have to go. We have so many students going this year. They really need a third. I'm sorry." And now they're old enough that he's like okay with handling them. He's like,
Yeah.
so there's 3, and we'll be gone for like a whole weekend and plus 2 whole school days.
There were only like 30 of us.
Like, I like something going right.
I'm so excited.
There's a lot. It was
Okay, so anyway, I know, I'm not even and I went, it rained the entire time. We didn't go on a ghost tour, and at Fenway, we got lost going to Fenway. It rained on us the whole way there. When we got there, we were soaked, and then it rained. I mean, yeah, I know.
Yeah, I know the weather's okay though, I'm excited.
We do a lot of walking.
That's okay. See, I don't feel like when I'm like big things, it's not like it's fun.
What you do?
Like no one's like that. The only time I went out was far was we had like my little cousin with us, and they were not about to walk a mile. Like they were
Okay, sorry. I digress. That's my pants.
I know.
Right, so there's 2 major types of cells, prokaryotes and eukaryotes, okay? Prokaryotes are what we call generally bacterial cells, okay? There's different types, we'll talk about them later. And then eukaryotic cells are everything else. So the major difference, like I said, all cells have these things. So prokaryotes and eukaryotes have all these things. The major difference is that prokaryotes do not have membrane-bound organelles. Eukaryotes have membrane-bound organelles. So one of those membrane-bound organelles is a nucleus. So the reason why prokaryotes got the name that they do and eukaryotes have the name that they do is because of this nucleus. Pro means before or pre. So in karyo is sort of analogous to carry on, which means kernel, and that would be like a nucleus. So before the nucleus is a prokaryote. So there is no nucleus in a prokaryote. The DNA is there, but it's located in the cytosol in an area we call the nucleoid region. It's kind of just in an area, but it's not bound up in a membrane. A eukaryote, U means true. Again, karyo, like kernel. So true nucleus. So they actually have a membrane-bound nucleus. The DNA is completely separated from the cytosol in eukaryotes. It's held up within the nucleus. Y'all remember this from Bio 1? I talked to Pablo this morning and he was talking about something. He's like, "Do they remember this?" I was like, I don't know. He said a lot of things.
I love Mr.
All right. Now this picture is misleading because it looks like parakaryotes are the same size as eukaryotes. That is not at all the case. Parakaryotes are much, much, much smaller. If you look here, 0.1 to 10 micrometers. They're teeny tiny little things. Eukaryotes could be 10 to 100 micrometers. They're much larger. Okay. So parakaryotes, here we go. So here would be a typical parakaryote. This is what it looks like under a TEM, transition electron microscopy picture, or even just a light microscope, I guess. But here we have the cell membrane in here somewhere. A lot of times parakaryotes actually have a cell wall. The bacterial cell walls are very different from plant cell walls. They're made up of peptidoglycan, of this different stuff. The bacterial cell walls we'll talk about in another chapter, but they can have them. They're just not the cellulose cell walls. You see the ribosomes hanging out in the cytosol, and you see the DNA here is all kind of coiled up in a region, but it's not in a organelle or in an organelle. Parakaryotes can have flagella or they can have cilia. These aren't cilia, these are pili that help them in what we call bacterial sex. It's called conjugation. We'll talk about that in another chapter as well. But they're not cilia. They don't function in locomotion. The flagella do. And here's another picture. Okay. We already talked about this. I usually like to draw a chart. So that's what I'm going to do right now, just to make sure that you know the differences between pro and eukaryotes. So let's just do a new slide. And I'll draw it for you. You get the difference, yeah? Okay, so let's see. Let's do examples first. So let's answer examples. So let's see. Have you heard of domains? Phenotype, class, source, and genus, species, but above that is domain. Yes, there's 3 domains. Bacteria, archaea, or archaebacteria, and eukarya. Okay, these are 2 different domains. Evolutionarily, they're not closely related. Archaebacteria, these are the extremophiles, the ones that live in like extreme hot temperatures, extreme salty conditions, stuff like that. Talk about those later. Eukaryotes would be everything else. So these would be your different kingdoms. Kingdom Protista, Plantae, Fungi, Animalia. Remember those? Yeah, okay. Unfortunately, in biology, they've kind of deleted a lot of the kingdom overview, which is unfortunate because it's actually very interesting. So I do talk about a little bit of this stuff as we go through, but not a lot. Anyways, okay, so those are examples. So we talked about size already. So eukaryotes are much larger. Location of chromosomes. So where was the chromosomes in prokaryotes? In the cytosol.
Breaks down the organelles.
anything that's bound by a membrane. So all the organelles basically are bound by the bilayer. Golgi apparatus, ER, chloroplast, mitochondria, nucleus. Ribosomes aren't membrane bound.
That's why there's-
Prokaryotes do have ribosomes, so do eukaryotes. That's why I had those on like all cell types. Okay. Good. All right. Talked about all this. You're good? Okay. Already talked about all this stuff. So eukaryotes do have membrane-bound organelles. They have all this other stuff that we will talk about. So here's our prokaryotes and our eukaryotes, plants and animal cells. So there's two types of eukaryotic cells. They look very differently, and they have a couple of different organelles. Notice both of them have this thing. What's that? Mitochondria. I told you this before, right? Student of mine, the day before the AP exam. Plants don't have mitochondria. Oh, yes, they do. Grant.
Oh, was that tall, Grant?
Grant like?
I don't know. Really tall?
Yeah, it was a long time ago, like in the old building. Like he's way out of college now.
We had a grant
It was like
when we
7
were,
feet tall.
when we were in 7th grade on the swim team. And I've always tried to figure out his last name.
7th grade.
He went to like either the Citadel or BMI. I don't remember.
Oh, okay, I don't know. Because Grant swam. He had a bunch of sisters, Elizabeth and Genevieve. They all went to Blythewood there.
All right, he was like 7 feet tall. Like he was.
No, that's not that Grant. He wasn't that tall.
Or well, maybe not 7 feet.
He's an example of someone who was fit in high school and he gained like
Is this
a
a
freshman 50. He stopped swimming and he gained a lot of weight. Poor Grant. I love you, Grant, wherever you are. He's very sweet, but he needs to start swimming or something. Okay, so why do we have organelles? Organelles are advantageous because they allow for specialization. They allow for certain things to occur in like compartments or certain areas. I mean, can you imagine real fast living in your house and there were no walls? Like your room was just like over there in the corner. Would you like that? No.
Yes.
It's advantageous to have compartments.
My room and my kitchen, everything's all the same place.
Oh, no. No, go to college. When you go to college, it's all there. And you're like, I just want my own space. Okay, no. Same kind of deal, okay? So you allow for specialization, right? You have a kitchen, right? You just cook in there. You don't cook in your bedroom. Next year, or when you go to college, you might. Anyway, okay. So there's specialized functions. So the cilia and flagella, they're made up of a lot of microtubules in a certain fashion, which we'll talk about. But they're specialized for cell locomotion. Some of them are like container type things. So the vacuoles that we talked about, that they talked about yesterday, the central vacuole of plant cells can store all kinds of stuff, sugars and toxins and pigments. Anyway, and they also can allow for a local environment that can have like a different pH. A lysosome is very acidic. Lulu will tell us tomorrow, but they have a very low pH, kind of a pH of like your stomach, okay? So it allows for digestion to take place, but the pH of the entire cell is not like that. So it can have a little bit of a different local environment. And they have very distinct functions. And I talked about that. So sometimes membranes themselves could be sites for chemical reactions. So in the mitochondria, mitochondria has a double membrane. The outer membrane is smooth and the inner membrane is highly folded and they're called cristae. And there's lots of specialized enzymes and proteins in that inner membrane that allows for a specific function for that to perform cellular respiration. Same thing for the chloroplast and the thylakoid membrane that they were talking about today. What's wrong?
She goes on that.
No. Today's October 1st. If you know Jay, it's his birthday.
Yeah.
It's Jim Hopper's official birthday as well today.
Oh,
I found that out, I was like,
That's funny.
that's pretty funny.
I love October. Pumpkins and fall and okay. Hold on.
I thought that was funny.
I'm gonna fast forward through a lot of this stuff. Hold on. All right. I'm gonna talk about this now. Sorry, Lulu. Okay. I'm gonna go through a brief overview of all the organelles. I'm gonna spend a little bit more time on some because I want to talk about specific things. Lysosome is one of the ones I'm gonna spend a little bit more time on. Lysosomes are only found, you also, by the way, need to know these organelles are only found in some are found in plant and animal cells, some are only found in plants, some are only found in animal cells. Lysosomes are only found in animal cells. And it makes sense because they're the site for digestion of food particles. Well, plants make their own food. Now, it doesn't mean they don't break them down and make other things. They do. They don't need lysosomes for the digestion of large molecules. Animal cells do because they're heterotrophs and they can't make food. Does that make sense? So it kind of makes sense that they do. But anyway, so lysosomes are what the little stomachs of the cell. They're the place where macromolecules are digested. Also, lysosomes can actually engulf organelles that are damaged or don't function anymore and help to recycle the components so the cell can use it again. So down here at the bottom, you see a lysosome is fused with a piece of the mitochondria, and it's digesting it, and the cell will recycle that stuff and use it again. This is the guy that discovered lysosome a long time ago, even before I was born. So,
Way before my parents were born, so that was really
how old are your parents? I'm sure your parents.
63 was.
Yeah, well, 74 was when its paper was published. So, yeah, my parents were born in 70. I was born in 79, so.
My sister was born in 79.
What? You're not old. It makes me feel old. Especially the other, I was like, he's like 40 something. Like, that's not that far from me. Oh.
You're not old, it's 5 years.
What?
Y'all were like,
You look very young.
Okay.
So, I mean, I'm all, yeah, but her kids are.
Wow, your mom's 60?
I did not believe that. My mom did not believe that. I keep
Anyway, okay. So let's talk about how lysosomes work. Lysosomes actually, I lied, they're not pH 2, they're pH of 5, but that's still relatively acidic. They make their own pH. I want to talk about how they do that. What they do is they have proteins in their membrane. Lysosomes in every organelle is bound by a lipid bilayer. Okay? So in that lipid bilayer of lysosomes, there are special proton pumps, is what they're called. You know what a proton is? A hydrogen ion, right? They're called proton pumps. They use the energy of ATP to pump hydrogen ions inside the lysosome. The more hydrogen ions you have, the lower the pH, right? The pH of the cytosol in most cells is around 7. So 5 is relatively acidic, yeah? Okay, so it creates its own pH by using ATP to pump in hydrogen ions against their concentration gradient to make it a little more acidic. So the enzymes that are in the lysosome have an optimum pH of around 5. So they work best there. 7 will actually denature them. One of the reasons this is advantageous for animal cells to have it like this is that what happens if you had one lysosome like rupture inside of your cell? The enzymes will denature and then you won't self-digest the cell, right? Okay, that would be bad. So it's a way to protect the rest of the cell by having it set up this way. Just like your stomach. If you got a rupture in your, if you have a rupture in your stomach, you probably have other bigger issues going on with you, but you wouldn't self-digest yourself because your enzymes would be, your pepsin would be denatured, right? So you wouldn't kill yourself that way. Now, like I said, there's other issues going on, but.
I don't remember.
I know exactly
She
what you're
had
talking
her sister had a hole in her heart. She had a hole in her heart, but her sister's stomach ruptured. It like, I don't know, she had a hole in her heart. She had a hole in her heart.
People can have a hole in your heart and not be. Anyway, okay. So that's good. Sometimes, however, lysosomes, you have a lot of them in yourself. Sometimes they can all rupture at the same time. And if that happens, you actually could digest yourself. And it actually is supposed to happen sometimes. It's called apoptosis. Have you heard of it?
Yeah.
Programmed cell death. It's when cells are supposed to die. They kill themselves.
They try using that for like cancer or something.
Yeah.
Like
We'll talk about it.
Get cancer cells to work.
We'll talk about the development of cancer and all that kind of stuff when we do mitosis, and we talk about it more there. But that's how you spell apoptosis, okay? Apoptosis is programmed cell death. It's when a cell actually commits suicide, and it's supposed to do it. And it does it for a number of reasons. One, it's one of the ways actually your body helps protect itself from going haywire. So cancer, when you develop cancer, when one develops cancer, a lot of built-in checkpoints have been just overridden, and a lot of things have gone wrong. Okay, but there's a lot of checkpoints that your body has built into place to help prevent cells that have abnormal DNA from developing into cancer, and apoptosis is one of them. The normal cells, when your DNA has been damaged beyond the point of repair, well, actually they're supposed to kill themselves, prevent themselves from dividing. Okay, so that's one way that apoptosis works. And how it happens is that there's actually a lot of different things. You may know Ann Visticonda. He has an older brother named Anurag, who's actually a first-year medical student at Columbia this year. He went to Yale. Well, Anurag was in my AP class a long time ago. He sat right here. And he did research at the School of Medicine while he was here in Discovery, and he worked on apoptosis, is what his research was on. But it was really cool because when I taught, we do a whole other section on apoptosis later on. There's a lot of different parts of it, but he studied a certain part of the mitochondria that's involved in apoptosis, which is really cool. Now I have to text Anurag and tell him. He does really cool dancing. He's on the Bonder team, right?
I know.
He's awesome. Oh my gosh.
Yeah. He's like what everything.
He was really good friends with Eric and oh gosh, I see his face. He passed away. About a year and a half. He just graduated from Rice. Oh my gosh. His name's gonna come to me later today. I'll be like, oh. Anyways, he had a stroke. No, was it a stroke?
Oh, I think I heard about that.
Wait, was this my friend?
Yeah.
Oh, yeah.
Oh gosh. Okay, I see his face. He was like Humans of Rice. He took all those pictures. He went to Rice. Oh, and he had a little sister, and they were like this close. It's really sad. And when he was here in Discovery, like his little sister was like 3. So cute. Oh my gosh. I'm sorry. Not Soria Vitamin. Soria Avali. Thank you. Yes. How do you look up?
Humans with rice dying.
Oh. Isn't he cute? It is. He had an aneurysm. That's what it was. It's really sad too because one of his best friends, one of Anurag's best friends, is kid named Eric, and Eric's dad passed away of an aneurysm when Eric was in 8th grade. Anyway, like really sad. Okay, Also, it happens when you are developing. So, you know, early on during fetal development, your fingers and toes are webbed. Did you know that? Right? So why don't you have webs anymore? Because of apoptosis. Most cells that make that up early on in development are programmed to kill themselves. And when it doesn't happen, people are born with, it's not like they have webs, but their fingers or toes are actually attached like this. And they actually can have surgery to separate them. But sometimes it doesn't happen. They're born like this, or their toes are like together. But it happens in development where they separate those cells undergo apoptosis to form your functioning fingers and toes. You can see it here and here where it didn't happen. But you can have it fixed surgically. So it's called syndactyly, yeah. But just the cells didn't like kill them, doesn't undergo apoptosis like they were supposed to. So it also happens like tadpoles when they're developing have a tail, but it goes away. And I already talked about the cancerous cells. So apoptosis is the thing that's supposed to happen. And lysosomes are involved in that. There's other things too. Okay, so sometimes things can go wrong. So there's a lot of different, I think over 40 different enzymes that can be found in lysosomes, a lot of different ones. And they function to break down different types of macromolecules. So what happens when one of those doesn't work? Okay, so when they don't work, that material cannot be digested, and it can actually build up and build up and build up to such a point that it causes the lysosome to burst, and it builds up in the cell, and cells will die, and eventually it could actually kill the organism. They're called lysosomal storage diseases. Like I said, there's over 40 known diseases from different enzymes that are known. Tay-Sachs disease is one of them. You might have learned about it in Biology 1 because usually they teach about it when we talk about genetic disorders. Tay-Sachs is a autosomal recessive disorder, meaning it's on one of your autosomes. It's not a sex-linked disorder. Recessive meaning you only get it if you have 2 non-functioning copies of the gene. Meaning you cannot actually synthesize that enzyme. It doesn't work. So it's a particular type of lipid that builds up in the nervous tissue, the central nervous tissue, the brain of these people, and the kids unfortunately die usually by the time they're 5 years old. It's a really terrible degenerative disorder that no one ever wants to have. A couple of years ago, I think we did a big thing for saving Eliza. I don't know if you remember that. We'd raised money for her, but she has something called Sanfilippo syndrome type A, and it's another one of these lysosomal storage disorders. She's really cute. Have you heard? Does anybody know who I'm talking about?
Yeah, we have
Yeah. A lot.
That's like a book for our house, like a photography book or something that they did as a fundraiser for Thomas.
Yeah,
It's really nice.
that is really nice. Eliza O'Neill is her name.
Oh.
She's been on like the Today Show and all kinds of stuff. What? Oh, not right now.
Yeah.
Anyways, this is Eliza right here. So these are a bunch of other kids now. They've made this foundation called Cura Sanfilippo Foundation, and these are a bunch of different kids, but this is Eliza right here. So I have to tell you the story, actually. There's a long story with this. It was a couple of years ago after I was on maternity leave with Marie, my little one, and I was actually at Panera downtown with my friend Katie, and I had Marie in like the car seat. She was sleeping. We were sitting having dinner. And Katie and I were sitting there talking, and this guy came up to us, and he was looking kind of scruffy. And he came up to us, and he was like, "Would you mind watching this video thing?" I don't know. We were like weirded out. We were like, we were just like, it's okay, we'll pass. Like, it was just awkward. And he didn't go to anybody else, and it was like, God, he didn't go to anybody else, and he just sat back down. And anyways, later on, I found out it was Eliza's dad, and he was working on this video that he first made to like do this. They had just found out that she had this genetic disorder. It was terrible. And I felt awful and I was like, oh my gosh. And I thought, Thomas, I was like, Thomas, we have to do something. I'm a terrible human. Anyway, but he made a video. It was the first one. You could find it somewhere. But Eliza's still alive. They tried to raise money for trials that were happening at Nationwide Children's Hospital in Ohio to fund clinical trials to do stuff to help cure Sanfilippo. Sanfilippo is a genetic disorder that doesn't affect very many people, and so there's not a lot of funding for it. Unfortunately, there's a lot of disorders that are not very common. There's not a lot of funding, so scientists can't work on them a whole lot. It takes a lot of money to do the research. Well, they raised lots and lots and lots of money. I don't think she fit the bill for the trial, so it's helping other kids. The family, the mom's a pediatrician, that's the mom in that picture there. And the dad works from home, there he is. He shaves enough, if you're not. Anyway. And they have a little boy too, but they've been in self-quarantine for over, I don't know how long it is now, but her condition will deteriorate if she gets sick with even just a cold or something. So they've been quarantined in their house for like a long, long time now, over like 200 days or something. Maybe a year. I don't know how long it's been. They have it shipped to sent to their house. Like people will deliver stuff and leave it at the front step. They'll go play in the backyard, or they'll go like to the beach or something where there's nobody there. They let her out, but they just don't have her around other people. They've gone to Nationwide, I think, the Children's Hospital, but she wears a mask, and they really limit her exposure to other people. That's their whole family. So anyways, they've been on the Today Show. They've been all over the place. But it's really sad because the problem with this disorder is that every day she lives, there's stuff building up in her body. And eventually it will cause her to lose functionality, like ability to speak, ability to move, ability to walk, and then eventually it causes death. I mean, you can imagine it's a terrible disorder to have. The child is one of these things because they're normal, and then they just do that. You know, and it takes a long time. So it's really, really sad. A lot of times with Sanfilippo, they can live to be like 13 maybe, or something like that. I think she's like 5 or 6 now. She was 4 when they found out she had it. This is an older video. Oh, okay. Anyway, so sorry, y'all are really quiet now. That was sad. They're from Columbia. They remember Miss Wet?
Yeah.
She lives right behind them. Yep. They are Columbia. They live in the Northeast. Anyway. It's really sad too because when they found out in one of the videos, the mom was talking about when the doctors told them that that's what Eliza had. She's a pediatrician, so she knows. She knows. So when she found that out, you can imagine just all of a sudden went through her head. If you're ignorant, not meaning ignorant in a bad way, but if you just don't know what something is, when the doctor first says, well, your child has Sanfilippo, you don't know what that means at first. You know, then the doctor really has to explain to you what that really means, but she knew immediately, basically death sentence. It's awful. Anyways, okay, moving on. Let's see what else is in here. Mitochondria chloroplast. Okay. So we'll talk about, sorry, that was a bummer. Mitochondria and chloroplast, and then we'll be done with the class for today. These are 2 other organelles. Mitochondria in all cells, all prokaryotes and eukaryotes have mitochondria. I mean, wrong. All plant and animal cells have mitochondria. Prokaryotes don't. They can still, well, we'll talk about what they can do later, but all eukaryotes have mitochondria, not prokaryotes, sorry. Only plant cells have chloroplast. So these organelles, the reason I have them on the same slide here is because they're sort of similar. They both have 2 membranes. Well, chloroplast kind of 3. But they're both transformers of energy. So they both actually make ATP. What they do with it is different. They both have double membranes. And they're both what we call semi-autonomous. What that means is that they kind of hang out on their own in the cell. They can both divide on their own. They both have some of their own DNA. And they both have ribosomes. And they can transcribe and translate their own DNA. And that thing that you watched last night, I hope y'all watched that video, that new research shows that the ER is involved in mitochondrial division. Did you see that? So cool. Yeah. Your book talked about endosymbiotic theory in this chapter, didn't it? Did it? How mitochondrial chloroplasts were probably evolved from free-living prokaryotes a long time ago? Did your book talk about that in this chapter?
I
You don't recall?
I read it somewhere online while doing that project.
Okay, well, we will talk about it now. Well, good, we will talk about it. Oh, maybe they didn't. Okay, so I'll end the class on this with what I just talked about. Do I have it? Okay, endosymbiotic theory. Let's spell it. You should know this. You have to know this. We'll talk about it again later when we do evolution, but let's talk about it now. This has to do with the origin of mitochondria and chloroplast. What does endo mean?
Outside. With their.
What does symbiotic mean? We live together. Okay, endosymbiotic theory. Something living together with something else inside of it. Okay? So long ago, the first organisms, the first cells on the planet were prokaryotes. No membrane-bound organelles. And they're all living together. Okay? Some of them would go and engulf another prokaryote to eat it. Okay? So what if, like, I'm the prokaryote that eats you, and you are very good at using the energy from the sun to make your own sugars. Okay? So I eat you, but all of a sudden, like, you're living in me, and, like, you're making sugars, and I'm like, whoa. You're making sugars. I don't have to eat this. It's making sugar for me. I don't have time to do it anymore. What? And I don't dissolve you. I don't digest you. You just start living together. And it gives you an advantage.
Question, varsity football players, reminder, please report to the varsity locker room at the beginning of lunch. Also, student tickets for the one ticket per ID holder for tonight's game. Justin Bowler, please come to the main office.
This is the rap battle.
Thanks, Andy. Anyway, okay, so they started living together in a symbiotic relationship, okay? And then over time, that evolved into a chloroplast inside of the prokaryote, which turned into a eukaryote because it no longer didn't have a membrane nowhere else. Anyway, so that's how they think mitochondria and chloroplasts actually evolved. We'll talk more about it tomorrow. The word theory means it's been so highly supported with evidence, we basically consider it definitely true. Got it? It's not just hypothesis. Theory means, like, in science, theory is like books of evidence. This is the lady that came up with endosymbiotic theory, Lenny Margulis. So I'll stop here. We'll talk about more tomorrow. Read chapter 7, take notes. Huh?
It was canceled before this happened.
Why?